subscribe, give lots of feedback, because I could use feedback. And mainly the reason why I'm doing this is I just want to, it's more fun to study this way. I want to share it, and it keeps me motivated. So, it keeps me focused too. Like, if I'm doing this streaming in front of people, then I'm not going to be getting sidetracked. So this is good for me, and I'm hoping it's good for you, because I know there's nothing I'd like more than watch somebody just going over concepts that'll prepare me to be a better doctor or prepare me to be a better scientist. I just feel like that's important, and I feel like we don't have enough fun with this stuff that we work on, the stuff that we study, and it's important to share community. I mean, I feel like we don't study group as much as we used to. I think back in the day people would study group a lot more often, and you kind of, in a sense, you don't really need to anymore, because you have all these extra study aids, and forums and etc. But I think the community aspect of studying can be really motivating for a lot of people, and I know it is for me. So without further ado, let us get more into the cellular receptors. Okay, let's see. Receptors for lipid-insoluble peptide, or what was this again? Was it catecholamines? Because I want to say hydrophilic, lipid-insoluble peptide, because this is not an positive. This is, right, catecholamines, yes. Okay, on the cell surface plasma, why is this so specific? Receptors for lipid-insoluble peptide, or catecholamine hormones, are on the cell surface plasma membrane. Is it that, I think, are catecholamines lipid-insoluble, or are they lipid-soluble? I cannot remember the structure of them right now. Let's see, they bind to the surface, because they bind to voltage gated. I have no idea if they're lipid insoluble or not, but that might not be relevant. So I assume that lipid insoluble, they tend to bind to GPCRs, and we had examples of that because we had large proteins and amino acids, things like those bind to GPCRs. Okay, so alright, so moving on. So glutamate would probably bind to a GPCR, but I'm not sure about that. Okay, ligand binds. Okay, so this is the first transduction pathway that we looked at for GPCRs. Ligand binds, receptor activates. Then that alpha beta gamma trimer attached to the G protein receptor, and then a GTP, a change in conformation, so that a GTP could knock off the GDP. Substitute the GDP, and that initiates the pathway. Ligand binding results in the binding of G protein right to the receptor to catalyze GDP. All right, cool. So we kind of covered that. Yes, G protein complex. Cool. All right, G protein dissociates from the receptor. All right, the alpha GTP and the beta gamma delta subunits dissociate. Yeah, both the alpha and the beta gamma can now interact with their appropriate effectors, E1 and E2. Now, I'm not sure if the beta gamma does E2, but I'm pretty sure that alpha does E1. And there's another variant where there's an alpha and there's a Q. It's alpha Q, and instead of interacting with the E1, that one interacts with PLC, which I'm not sure what that stands for. Alpha catalyzes the hydrolysis of GTP to GDP again. Oh, right. No, this one stays on. Inactivates alpha and promotes reassembly of the trimer. Cool. Okay, cool. So that tells me that happens. That's actually really useful. Yeah, and that change in conformation allows it to attach to E1. Okay, no, yeah, and then E1. Okay, yeah, gamma beta does E2. I wonder if this is involved in the muscarinic, because there's an M1-2, something like that, receptor, and that definitely requires modulation via gamma beta gamma. So we'll have to look into that later. All right, ionotropic receptors, ligand gated ion channels. Okay, cool. So an example is the nicotinic receptor, or nicotinic acetylcholine. And we know that nicotine is cholinergic. Okay, moving forward. Catalytic receptors have an extracellular domain. Ligand binding happens to the right. Just blah blah blah. All right. Moving on. We'll learn more about that, because that card has a whole bunch of extra details. That note has a whole bunch of little cards in it. Okay, GPCR activation. Effector molecules pathways for All right, GPCR, beta gamma subunits, modulation of effector proteins. Okay, GPCR, beta gamma subunits, were originally described as simply anchor proteins, yes, but now we know that they attach. Right, so this is the one, the M2R receptor, the muscarinic 2 receptor. Increase calcium or potassium channel activity via G beta gamma. Oh, so that's the difference between muscarinic and nicotinic. So nicotinic allows sodium to go into the cell, hyperpolarizing it. Muscarinic receptors, on the other hand, depolarize. So that is interesting. That is interesting. Yes, modulate signaling, like we talked about before, which I think is the first one. Okay, then we have 2, and this actually has 2 and 3. This is the PLC one, where the alpha subunit, and this one's alpha Q, will interact with the PLC protein, which then interacts with the PIP2 protein, and causes inositol 3-phosphate to go to the endoplasmic reticulum and release calcium. That then facilitates the binding again with the alpha beta gamma, or alpha Q beta gamma. But the PIP2 goes activate. There's a DAG, and the DAG then binds to the protein kinase C. So those should be the 2, and that one's the indirect pathway, and then there's a direct pathway that I should cover, and we have PLA. So instead of pressure liquid chromatography, in the direct pathway, we have the PLA plastic, phospholipase A2, with a phospholipid and arachidonic. So right, the DAG, and in the indirect pathway, the DAG activates the MAG. Okay, whatever. Instead of the protein kinase C or whatever. So we have really 4 actually, unless MAG is a type of protein kinase C, PKC, which no, it's not. Anyway, all right, so we can have 4 different pathways here. I should really remember. Oops. Well, yeah, okay. I mean, I'm willing to accept not getting that one right. Okay, sinus, nasal cavities, sinuses, larynx, trachea. Yep, that's an easy one. All right, general characteristics: guanyl cyclase, right? Catalytic receptors, ANP, extracellular domain. Wow, I did not read that. Okay, atrial natriuretic peptide. Yes, that is familiar. I do believe that is released. Is that released by the baroreceptor in the aorta or in the atria? No, wait, no, atrial. I'm not sure what that does. I'm not sure what that's all about. Guanyl cyclase, yes, and it does dimerize in order to work. All right, we're gonna need some more details about this. Otherwise, it's gonna be harder to remember. Look up. I do not remember from anatomy and physiology what this does. Okay, atrial natriuretic peptide, or atrial natriuretic factor, is a natriuretic peptide hormone secreted from the cardia atria. Natriuretic peptide is a family of hormones/paracrine factors that are structurally related. The main function of the ANP is causing the reduction in expanded extracellular fluid volume by increasing renal sodium excretion. Interesting. ANP is synthesized and secreted by cardiac muscle cells in the wall of the atria of the heart. Okay, interesting. Maybe this is a response to blood pressure? I still don't entirely understand this. Okay, we'll have to look into this more later. Share, make a reminder, please. I guess. Look up. Oops. Getting ahead of myself.